Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine a single medicine, something that could help your body fight off, well, not just one virus, but a whole range of them. Everything from the common cold maybe to something more serious like Zika virus or even herpes. It sounds almost, I don't know, too good to be true, doesn't it? But what if we told you that researchers have actually made a discovery that's pushing us closer to exactly that kind of reality?
That's absolutely right. Our deep dive today is looking at really a groundbreaking breakthrough. It's from researchers at MIT and some other institutions, and they've identified a whole new class of compounds, and these don't attack the viruses directly, which is how most antivirals work. Instead, they they supercharge your own body's natural defenses within your cells. It's a completely different way of thinking about antiviral medicine.
Okay, so our mission today is to unpack exactly how these remarkable compounds work. And why are they being called broad-spectrum antivirals? And maybe most importantly, what could this incredible discovery actually mean for the future of fighting infections? We'll be drawing from both a health news article and the scientific paper itself to try and give you the full picture.
Right. So to really get this, we need to start with something called the integrated stress response pathway. It's a bit of a mouthful, I know. Let's maybe call it the pathway or the stress response sometimes. Think of it as your cell's built-in universal defense system. It's fundamental, totally essential, and it's conserved. That just means it's been so vital for survival, like throughout evolution, that it's pretty much the same across almost all complex cells, from yeast right up to us humans. Its main job is maintaining balance, what scientists call homeostasis, inside your cells when they face stress.
So it's like the cell's internal security system, always on alert. How does it specifically react when, say, a virus invades? What's its main weapon?
That's where it gets really clever. When a virus infects a cell, it often produces this kind of unique distress signal. It's a molecule called double-stranded ribonucleic acid.
Okay, double-stranded RNA.
Exactly. And that's usually not found floating around in healthy cells, right? So the integrated stress response pathway spots it almost immediately. And in response, it essentially slams on the brakes for the cell's ability to make proteins.
It stops protein production. Why is that so important?
Because viruses are basically parasites. They need your cell's protein-making machinery to replicate themselves to make more viruses. So by hitting this off switch for protein production, the cell effectively blocks the virus from multiplying. It starves it out, in a way.
That really is a massive shift in thinking, isn't it? I mean, for so long, we've often been playing this endless game of whack-a-mole, right? Chasing each new virus with a very specific drug. This, though, sounds more like building a universal shield by just boosting our own body's defenses.
Precisely. That was the core idea the lead researchers had. They hypothesized that if they could find compounds that could modulate, basically boost or fine-tune this integrated stress response pathway, then they might discover a whole new class of antiviral drugs. Drugs that wouldn't be narrow just for one virus, but could potentially work against many different kinds. A true broad-spectrum approach.
Okay, now this next part, how they actually found these compounds, is genuinely fascinating. You mentioned they use light. How did they use light to simulate viral infection?
Right. This is their ingenious optogenetic discovery method. It's pretty cutting-edge stuff. The goal was to find compounds that specifically enhance that stress response pathway, but only during what looked like a viral attack to the cell. So they used this bioengineering technique called optogenetics. It involves inserting light-sensitive proteins into a cell's genetic material.
So they basically engineered the cells so they could flip a switch with light, like tricking them into thinking they were infected.
Kind of, yeah. It does sound a bit like cellular espionage. They specifically engineered a protein called protein kinase, or PKR for short, but we'll call it protein kinase R. It's a key player in turning on that stress pathway. They engineered it so they could activate it just by shining blue light on the cells. This let them simulate that viral infection signal without actually having any virus present. It gave them incredibly precise control.
Okay, so they had these light switch cells. And then came the massive screening process. This is where the scale just sounds enormous.
Oh, absolutely huge. They screened a massive library of nearly 400,000 different chemical compounds.
400,000. Just to imagine testing all of those.
It's a huge number. So Human cells, engineered with this light-switchable protein kinase R, were exposed to both the blue light simulating the infection and the different compounds, and then they watched the cells' survival rates. This allowed them to identify which compounds potentiated or amplified the integrated stress response pathway, helping the cells shut down that simulated viral reproduction signal. From that massive effort, they narrowed it down to about 3,500 candidates.
Okay.
And then further still to just a handful of top contenders.
And from that incredibly rigorous screening, they landed on 3 top compounds, right? Named IBX200, IBX202, and IBX204. What makes these particular ones so special, so promising?
Their uniqueness is really the key. These compounds, they selectively increase cell death, but only when the integrated stress response pathway is already activated, like by the blue light in their experiments or, by an actual stressor like a real virus.
Right, so they don't just harm any cell.
Exactly. This is a critical distinction. It means they don't seem to cause widespread toxicity to healthy, unstressed cells. That was a major problem with many previous attempts to activate this pathway. The drugs were just too toxic. That's why these are called integrated stress response potentiators. They boost the response when it's needed. They don't just turn it on full blast everywhere.
Okay, that makes sense.
Yeah.
So they boost our natural defenses selectively.
Yeah.
But what about actually using them as medicine? Do they have the right properties? Things like, can the body absorb them? Can they get to tricky places like the brain?
Yeah, crucial questions. And the good news is these compounds seem to have favorable drug-like properties. They are Lipinski conforming, which is a technical term, but it basically means they have characteristics suggesting they could be well absorbed by the body if taken as a pill, for example. And yes, their molecular weights also suggest they might be able to cross the blood-brain barrier. That's a huge hurdle for many drugs, especially for brain infections. Plus, they have novel chemical structures, which is always exciting for potential future drug development. It opens up new possibilities.
Okay, so they look good on paper. They work in these engineered cells. What does this actually mean for fighting real-world infections? You mentioned they show promise as a broad-spectrum shield. What kind of actual viruses did they test them against in human cells?
Right. They tested them against a really diverse range of viruses, which is important to show that broad activity. They looked at Zika virus, that's a positive-sense single-stranded ribonucleic acid virus. They also tested herpes simplex virus, type 1, that's a double-stranded deoxyribonucleic acid virus, so structurally quite different. And also respiratory syncytial virus, RSV, which is a negative-sense single-stranded ribonucleic acid virus, so quite a mix.
A real variety then. And the results, were they as exciting as hoped?
Absolutely. The results were very promising. Treatment with these compounds significantly reduced the amount of virus in the cells for all 3 types they tested.
Wow.
This is a huge deal because it strongly suggests they don't target a specific bit of the virus, which is what most antivirals do, and why they only work on 1 or 2 types. Instead, these seem to target that host pathway, the cell's own response, which gives them that potential broad effectiveness.
And how do they seem to work? Do they stop the virus getting in or stop it multiplying?
They appear to impair viral replication, so they stop the virus from making copies of itself inside the cell, rather than just blocking entry. It is worth noting, one compound, IBX204, was slightly less effective against the herpes and the RSV viruses. And the researchers think this might be because it didn't quite boost the levels of a crucial protein called activating transcription factor 4 as much.
Activating transcription factor 4.
Yeah, think of it as one of the key soldiers in the cell's defense squad, mobilized by the stress response pathway. If its levels aren't boosted enough, the full defense isn't quite as strong.
Okay, I see. But still, impressive results across the board. And this wasn't just in cells in a dish, right? They took it a step further into living organisms. That seems like a critical step.
It absolutely is. That's the in vivo testing in a living system. They took one of the compounds, IBX200, and tested it in a mouse model of ocular herpes virus infection, so herpes infection in the eye. It's a well-established model for studying viral infections.
And what did they find in the mice?
The findings were really excellent. Mice that were treated daily with IBX200 eye drops showed improved disease scores, so less severe symptoms, and they had a significant reduction in the viral load, about a 5-fold decrease in the amount of virus particles found in tear swabs.
A 5-fold decrease. How does that compare to existing treatments?
That effect was comparable to a known antiviral drug, acyclovir, which is often used for herpes.
Wow. So it worked about as well as an established drug, but through this totally different mechanism of boosting the host defense. That's really significant.
It really is. Plus, there was another interesting finding. The treated mice also showed lower levels of something called CXCL10IP10. That's a chemokine, basically a signaling molecule that's a marker of inflammation during herpes infections. So it suggests the treatment might also reduce inflammation, which is an added benefit.
That is impressive. It really makes you wonder about the potential here. But of course, whenever you talk about a potential new medicine, the very next question has to be about safety, right? What did their initial toxicology tests show? Are these things safe?
A critical aspect, absolutely. And the initial toxicology profile for these compounds looks quite favorable, which is very encouraging. Preliminary lab tests on IBX 200, 202, and 204 showed they were not hemolytic, meaning they didn't damage red blood cells. They didn't chelate iron, which can sometimes cause problems in the body. And they weren't genotoxic. They didn't damage DNA even at relatively high concentrations, around 200 micromolar.
Okay, that's good news from the lab tests. What about in the living mice again? Any signs of trouble there?
Also promising results there. In mice, when they injected these compounds at a dose of 50 milligrams per kilogram, they didn't see significant increases in key liver enzymes, things called AST and ALT. Rising levels of those enzymes are usually indicators of liver damage. And they compared this to a known toxic drug, doxorubicin, which did cause those enzyme levels to spike. So by comparison, these compounds looked much better in this early test.
That initial safety data sounds really encouraging then. So stepping back a bit, how are these compounds actually doing this? What's the deeper molecular mechanism? How do they supercharge our cells' defenses?
Right, getting into the nitty-gritty. The core mechanism seems to involve another key protein. These compounds appear to boost the integrated stress response pathway, at least in part, by increasing the active form of a protein called general control non-repressible 2.
Okay, general control non-repressible 2. Another mouthful. What does that one do?
You can think of general control non-repressible 2 as maybe a master control switch or a precision instrument within this whole pathway. When general control non-repressible 2 gets activated, it leads to increased production of those crucial downstream proteins, like the activating transcription factor 4 we mentioned earlier, and another one called CEBP homologous protein. These guys really orchestrate the stress response.
Okay.
Now, in a healthy cell just facing minor stress, activating general control non-repressible 2 might just lead to an adaptive response. The cell adjusts and carries on. But, and this is the crucial part, when a cell is seriously under attack, like from a virus, and the stress levels are high, fully activating general control non-repressible 2 seems to push that infected cell towards a terminal response.
Terminal response, you mean?
Essentially self-destruction. Programmed cell death. It eliminates the infected cell, taking the virus with it and stopping the infection from spreading to neighboring cells.
Ah, I see. So the compounds kind of push it over the edge, but only if it's already seriously stressed by the virus.
Exactly. That seems to be why they're selective. They amplify the signal when it's genuinely needed for a strong defense, rather than just triggering alarms in healthy cells.
It's a really elegant biological mechanism they're tapping into. But like all research, especially early stage, there must be limitations or areas needing more work, right?
Absolutely. No study is perfect, and there are always next steps. For instance, going back to that optogenetic tool they used for discovery, while it's incredibly powerful, the blue light itself could potentially have other subtle effects on cells, maybe inducing some low-level stress or generating reactive oxygen species.
Right.
The researchers did take precautions and run controls, but, more detailed control experiments are always valuable to completely rule out any off-target effects just from the light stimulation method itself.
And beyond the tool, what else needs more exploration before we can really know how useful these might be?
Well, even though they showed broad activity against 3 different virus types, their effectiveness still needs to be rigorously tested on a sort of disease-by-disease basis.
Yep.
We need to fully confirm their broad utility in many different real-world infection scenarios.
Makes sense. One size might not fit all perfectly, even with a broad approach.
Exactly. And we also need more detailed studies to fully map out the precise molecular interactions. How exactly do these compounds bind to and activate that general control non-repressible 2 protein, for example? Getting crystal structures, basically detailed 3D pictures of the compounds attached to the protein, would give us much clearer insights into how they work at the atomic level.
And I suppose the ultimate goal is always moving from the lab bench towards actual clinical use.
Absolutely. That's always the long-term aim. The next phases of research will likely focus on identifying even more compounds, perhaps related to these ones, or refining the existing IBX compounds, trying to make them even more potent, more effective, maybe tweaking their properties for better delivery in the body, all with the ultimate goal of eventually moving towards human clinical trials. It's definitely an ongoing journey, step by crucial step.
Okay, so as we start to wrap up this deep dive, let's bring it back. What does this all mean for us, for the listener? We've explored this really exciting potential of what they're calling integrated stress response potentiators, this new class of broad spectrum antiviral compounds that leverage your own cell's defenses. And as you said, this isn't just about finding another drug. It feels like a whole new strategy for fighting viruses.
I think that's the key takeaway. If we connect this to the bigger picture, you can start to imagine a future where maybe we have access to a more universal type of medicine, something that can help our bodies respond effectively to a whole variety of viral threats, whether that's newly emerging pandemics or persistent infections like herpes that stick around for years. It could fundamentally change how we approach infectious diseases. Maybe, just maybe, reducing the need to develop a brand new, highly specific drug every single time a new virus pops up. It's about empowering our own biology.
That really leads to a pretty provocative thought, doesn't it? In this world that's so interconnected where we constantly face new viral challenges, what kind of impact could a truly broad-spectrum antiviral one that works with our body actually have on global health, on our daily lives? It makes you wonder, what kind of follow-up questions do you listening now have after hearing about this potential breakthrough? We definitely invite you to consider those possibilities and just appreciate this ongoing journey of scientific discovery that, well, it continues to shape our world in pretty amazing ways.
Thanks for tuning in to the Health Headliner podcast by Todo Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.